Thank you very much. First of all, I would also like to extend my gratitude for the organizing committee for having me, and thank you for keeping up attention today and to giving me the floor for this last presentation of this session, which is a bit different from the previous three speakers actually, because I would like to take the floor today to discuss with you the opportunities that European food law already gives to use alternatives to animal experiments. And I will explain you later on why I'm doing that here today. First of all, the issue with using animal experiments, well, most of you are in nutrition research, so I probably do not need to explain that a lot of regulatory procedures, but also science in general, relies on identifying toxicological issues, but also other potential food safety consideration and nutritional considerations based on animal experiments. And we believe that there are opportunities to reduce what we think is an over-reliance on animal data to maybe shift towards using other methods, newly developed methods, and maybe integrating these methods as well. And today my talk is focused on that. However, this is not only just my contribution. I would like to thank ILSI Europe and also the other expert group members of the task force on alternatives to animal testing, because this work that I present to you today is developed with 15 people in total with a range of different expertise to study this concept of animal-free strategies in food safety and nutrition. My disclosure, I do not have any conflict of interest next to having received a small compensatory sum from ILSI Europe for travel support and for conducting this work. And the expert group members are both from industry and different universities which have been grouped together to study this concept of animal-free strategies. Going back to the research aim and why I'm presenting here to you today. Our aim of the expert group was to focus on the opportunities that are there in mainly European food law, where we need to conduct lots of different scientific experiments to get pre-market authorization, to get authorization to be placed on the market for new products, and to review the possibilities that are already given, both from the scientific perspective and the regulatory perspective, opportunities, but also challenges for the potential to use non-animal testing strategies. Before I go into the details of our findings, I would like to give you some background information on that. Why do we even consider the reduction of animals? Why is that so important? In Europe, there's a specific directive that tries to encourage the reduction of experimental animal use. So the directive, which is called the Protection of Animals Used for Scientific Purposes, brings about the legal obligation for everyone in the field of science, not just food science, but science in general, to actively promote the implementation of the so-called 3R strategy: replacement, reduction, refinement of the use of animal testing. And Article 13, I want to point that out to you, describes that animal tests should not be carried out if any other methods for obtaining the results sought, not entailing the use of a live animal, is recognized under the legislation of the Union. That is literally from the directive. So this directive tries to make sure that if you have another method available to get to similar results, please use these alternative methods. Why do we focus on European food law? Well, European food law deals with everything related to food authorizations. European food law, to give you a little bit of background on that, has its basis in the so-called framework regulation, the general food law. And this general food law is mainly the framework regulation, as I said, and it lays down the general principles and requirements of food law. It's a real farm-to-fork approach to food law, so it does not only focus on specific sectors, but it tries to lay down the general principles for all players in this field. What is really important in this general food law is that the responsibilities for conducting risk assessment is separated from the responsibility to conduct risk management. So the scientific work is done by another institute than the political decision to allow a new product on the market. This risk assessment is EFSA's task, and the political decisions the political responsibilities lay mainly by the European Commission. In everything related to food law, scientific evidence is key. Scientific evidence has to underlie the political decision. So this risk assessment part is essential in any authorization decision. However, next to this framework regulation, there's also sectoral regulations and directives that deal with specific fields of food products, specific fields, for example, in hygiene measurements, safety assessment, or testing requirements. But their common denominator is, as I said already, there is a need for pre-market approval. Before you can place a product on the market, a new product, or you can provide a new claim on your product, you have to show the scientific evidence. The responsibility for that is on the industrial. They have to provide the scientific evidence. It's reviewed by the scientific authority, by the European Food Safety Authority, and the European Food Safety Authority provides their scientific opinion to the political decision maker. And that political decision maker decides whether they indeed approve the authorization request. Again, the scientific evidence is key in that decision. And we find many specifications on how to do that scientific research in EFSA guidance documents, not necessarily laid down in law, but often in the specific guidance requirements how to deal with this specific law. And when we look at the scientific developments in testing of foods, food safety, there is a very nice publication already by Blauboer and colleagues that already stipulate the potential to use non-animal strategies in not only food safety, but also nutritional considerations for risk assessment of foods. They propose to focus on the shift of the apical endpoints of toxicity, where we use clinical observations, for example, from animal models, to more exposure-driven approaches. And this is relying on understanding the mechanism of toxicity instead of focusing on these endpoints. There's a list of distinct aspects that are needed for safety evaluation, but for the sake of time, I'm not going to focus on that. You can read that in this very interesting paper by Blauboer and colleagues. But the focus of their paper is also to say this risk assessment for foods and the determination of safety levels can be done mostly without animal data. It's not always necessary to conduct new animal studies to gain insights for the risk assessment of a new food product or new food ingredients. So these elements were the basis to conduct our study, in which we reviewed different sectoral laws, different product categories that are regulated in European food law, and to study the opportunities that are already there to use alternative methods to animal experiments. So I want to start by showing you some sectoral legislation that deals mainly with food safety, and I'll come back later to the nutritional elements. So I'm going to give you an overview per category, and I want to focus here on what is called in European food law new nutrition sources, the food improvement agents, novel food, and GM foods and crops. But the basis, as I already said, is laid down by the general food law. And this general food law says all food should be safe. However, should be safe for the general population. So, of course, if your food contains an allergen, it's not necessarily the case that you're not allowed to place it on the market. You just have to take that into consideration. But the main element is that food should be safe for this general population. And all these different laws that I will address are based on that general idea, that general concept. So first, new nutrition sources. Well, that's quite a vague term, of course, but in European food law, it relates to food supplements or ingredients for food supplements, nutrients that are added to foods, for example, for fortification purposes, but also ingredients that are used in foods for special groups, for example, in infant foods or foods for medical purposes. And interestingly, even though I said that the general food law does not specify any data requirements, it's just lays down general principles. For these types of new products that are placed on the market, their authorization procedure is based on the General Food Law. But this article of the General Food Law only says EFSA can be consulted to review scientific dossiers. There are no data requirements given. So how should that scientific dossier look? Well, there is EFSA guidance, and this is from 2018, relatively new, that suggests to take a tiered approach which includes biological and toxicological data. But there's no more information than that, except for some minor data requirements, or minor data requirements that is just minorly mentioned. It is suggested to conduct bacterial reverse mutation assay, in vitro mammalian cell micronucleus test, and the modified 90-day toxicity test. There is no suggestion given for using alternative methods to, for example, this 90-day toxicity test. Moving on to these food improvement agents, I want to highlight two types of improvement agents where they are in European food law clustered as enzymes, additives, and flavorings. I will talk only about additives and flavorings here. These food improvement agents share the common authorization procedure for food improvement agents, but their data requirements may vary because the substances and their potential effects, of course, vary. In Tier 1, for the Tier 1 data sets, the minimum data set, it is described for food additives that animal testing may be avoided. However, if the results from Tier 1 trigger the conduction of additional testing, this is very often relying on animal testing, even though, as you can see from our analysis, that we have identified different opportunities where you can replace animal testing already by using mainly in vitro methods. And this especially goes for toxicokinetics, genotoxicity, and allergenicity. Interestingly, for food additives coming from a botanical source, if you use that botanical source already longer in the general diet, and there is a history of safe use, also this additive can use this type of data, as we call it, the presumption of safety. So you do not need to go through all the additional testing if you can show there's evidence of history of safe use. And EFSA describes in their guidance for food additives a clear reduction effort to reduce the use of animal testing. They suggest to different people and organizations who want to apply for a new food additive authorization to focus on reproductive toxicity studies, if you need to conduct that, to focus on the extended 1-generation study instead of using the 2-generation study, which would save many experimental animals. And also the grouping of additives to use read-across techniques within a group of additives is highly encouraged. This also goes for food flavorings. As you can see on the sheet, the last suggestion, also in the EFSA guidance, is this grouping approach. Also here, Tier 1 animal testing may be avoided, even though the 90-day subchronic toxicity study in rodents is required depending on the exposure. There is another reduction effort suggested by EFSA in their guidance that the threshold of toxicological concern, the TTC, will also be accepted. So if the exposure to a certain food flavoring is expected to be very low, this could lead to the realization that no additional animal experiments are necessary. Moving on to novel foods. In Europe, they are defined as new or traditional foods from outside the EU. So newly processed ingredients, but also traditional foods that could have a safe history of use in a third country, as long as they are not consumed significantly within the EU before May 1997. We have 10 categories. I will not address all of them, but to show you an example is, for example, very much related to the previous speaker, the use of insects, which would fall in the 5th category, foods consisting of or isolated from animals or their parts. So for these types of products, you also need to show with scientific evidence that they're safe before you can further apply for an authorization. The traditional foods from outside Europe can build that dossier based on history of safe use, but for novel products, it's necessary to show insights on kinetics, nutritional effects, the toxicological info, and allergenicity. Here also, in EFSA's guidance, the tiered approach is suggested. The 90 days of chronic toxicity study is normally required because the results may trigger additional studies. The read-across of toxicological data can be considered, and also the TTC approach is suggested. However, interestingly enough, this guidance on this, the guidance on this specific regulation originates from 2018 and builds very much on the food additive guidance that originates from 2015. And whereas the food additive guidance explicitly addresses the reduction efforts of EFSA, the reduction of animal testing efforts, in EFSA's guidance on the novel food regulation, it merely says that non-animal methods are accepted case by case and does not show any insights in how to use these novel methods. The fourth and last type of sectoral legislation that I want to address regarding food safety is that on GM foods and crops. There's a whole list of things you need to consider in your risk assessment. But the most important and most interesting element from our analysis is that there's one legal requirement. And I already showed you the most data requirements came from guidance documents, not from the law itself. But for GM foods and crops, there's actually the legal requirement to conduct a 90-day subchronic toxicity study in rodents. However, the scientific value of conducting such a test is highly questioned because the results, the adverse effects that are found in these studies are very rare if you have already gone through the other steps of risk assessment. Moving on to the nutritional assessment, because also there the use of animal testing methods is used or suggested in different guidance documents or based on the endpoints that are required in food law. And these include foods for special groups, where I focus on infant formula and medical foods, and for health claims. So if you think about infant formula, the main element to consider in the risk assessment is allergens. And the requirements that are laid down again in the guidance documents include the suggestion to study allergenicity in vivo by means of rodent models. However, the prediction potential for humans of these models is highly questioned. And we also see that there are scientific developments into establishing new methods, mainly in vitro models based on human cells, to study the allergenic potential of these different substances and to study the allergenic potential. For medical purposes, for medical foods, you need to prove the efficacy that this specific patient group actually needs your food product or needs your food ingredient in their disease management. So you need to show the relevance and the need in a patient group. And EFSA's guidance merely mentions that you need to provide kinetics data and data on the nutritional status of the patient, which is often still studied by means of using animals. Moving forward to the last example that we focused on in our research are health claims. And in Europe, it's stipulated that you need human data to support such a health claim. So you need to show that, first of all, of course, your ingredient is well characterized, your effect is well characterized and actually a beneficial effect to human health, and you need to show a cause and effect relationship. And this cause and effect relationship needs to be substantiated by human data. So why is this study, why has this been even part of our studies? Well, interestingly, there is no EU requirement for animal data to support claims, but very often local and national ethic committees, they demand animal data for potential efficacy before it's even allowed to start a human clinical trial. So this is quite, well, alarming, we believe, that even though it's not a regulatory requirement to conduct animal experiments, it is a requirement from local and national ethic committees. So to summarize what kind of methods we identified in the scientific literature, but also suggested in different guidance methods, are especially on top of the sheet, the suggestions to use alternative methods for in vitro genotoxicity tests, to use read-across and grouping, and to use the TTC concept. And it's very clear from the guidance documents of EFSA that these methods are already considered by the regulatory regulatory procedures to be sufficient, or at least add to the insights in risk assessment under certain conditions. However, we also see that there are way more potential methods available already. We see computational models, organ-on-chip models, also high-throughput methods for mechanism of mode of action assessments, and other uses of, for example, adverse outcome pathways. And we believe they have a big potential, but it's very unclear whether they are accepted already in regulatory dossiers or whether they are merely now used in science and need more validation of their potential use in regulatory dossiers as well. It is highly unclear whether these potential methods would fall into the case-by-case assessment within the risk assessment by EFSA. So what I hope you can take away from our research is that, first of all, EU legislation and guidance, we've seen that mainly these guidance documents give suggestions for how to do the research that is needed in regulatory approval processes. We, however, believe that these are relatively static, similar to the food law in general, and updates are necessary as soon as the alternative methods become available. We believe it's interesting and maybe even alarming to see that guidance in 2015 suggests the use of reducing animal experiments, whereas this is less stipulated in guidance documents from 2018. Secondly, assessing health benefits, the efficacy of nutrition, which is not even required to use animals in some cases. There, science should focus on building evidence and validating in vitro approaches to substantiate clinical trials in humans, to make sure we do not over-rely on animal testing here. And looking at the scientific literature and the regulatory approval procedures, we believe it's necessary to focus on the promising and available methods to avoid animal testing, to further stimulate and apply them. Thank you very much for your attention.